Hello ladies and gentlemen, welcome back. My name is Dylan Hamilton from C.A.R. Entertainment, and today we're going to be going through AS1 Biology, more cells revision. We took a couple of days off just because it's tracking week for some schools here. I know it is for my school, so basically trying to do a wee bit of revision for that there. But we're back here now. We're going to do mitosis and a wee bit of cancer here, and then we're going to do meiosis, and then we're done with cells. Then we only have tissues and organs, and so that includes the ileum and the leaf, and then we're done with AS1. Let's get into this then straight away. Basically, not much you really know about chromosomes, because that's going to be about that. Is going to be the first thing we're going to talk about. Basically, a chromosome is just a long strand of DNA bound to histones. DNA strand replicates to a new histone, and these two replicated strands remain attached to the structure called a centromere. So centromere is the little bit that connects the two chromosomes, so it's the horizontal line, or yeah, it's supposed to be a line whenever you see it. So then the cell will begin to divide, and each strand will coil and then supercoil until it's visible as a sustainable double structure, and they're replicated to two sister chromatids, and each chromatid is identical to a sister and an original chromosome. Chromatid is different than a chromosome, so a chromatid different from a chromosome. Now, the cell cycle. So the cell cycle includes a number of phases. We're going to start off with interphase, which is basically a two-gap phase, G1 and G2, and this is separated by a symphysis S phase. So mitosis, essentially, it's the nuclear division of chromosomal material partitioned into daughter nuclei. So daughter cells, that's where you get that from, not haploid, daughter. You'll get diploid, not haploid. So and at the very end, you'll get cytokinesis, where the cell divides into two daughter cells. So yeah, daughter cells will be produced, and they'll have the same chromosome as the parent cell. It's the division of chromosomes, cytokinesis, division of cell. Okay, so mitosis, division of chromosomes, cytokinesis, division of the cell. So, specifically, let's go through these stages then. So, interphase. G1 is basically your synthesis of macromolecules, and then your S phase will occur, which is where DNA synthesis will occur, as histones will bond and support the DNA in chromatids, with DNA and chromatids being formed identical to the remaining attached, and they will remain attached until mitosis actually goes through and takes place. Then G2, the G2 phase will occur, and proteins will be synthesized with tubulin, forming microtubules of the spindle fibers. Of course, we did talk about those earlier on in cells. So the DNA and cell cycle is the preparation for mitosis, and the interphase we just talked about. So the DNA of the cell will double, and the DNA will double during this S phase that we talk about, and then it returns to normal for cytokinesis, which is the division of the cell. Now, there'll be checkpoints that this will go through. So it is essential the cell will only divide when there's sufficient enough macromolecules assembled, so the cell will grow to an appropriate size, and the DNA is error-free. Now, there's a number of checkpoints that the cell will have to pass before it progresses. Basically, at the end of G1, the supply of nutrients and the growth factors will be assessed. You need to know if the cell is grown to an appropriate size, if any damage has been detected, and if the DNA is repaired, it will then proceed to S phase. If not, it will enter a resting phase. Then our second checkpoint will be at the end of G2, where the DNA will be replicated, and if it isn't, the cell cycle will be stopped, which means you can check for DNA damage during replication and the delays for repair. Now, the final checkpoint is when metaphase establishes chromosomes correctly attached to the spindle fibers before anaphase proceeds. We'll move on to what metaphase and anaphase is. Basically, if the chromosomes aren't correctly attached to the spindle fibers before anaphase, then mitosis will not go ahead. Now, let's talk about cancer for a wee bit, because this is basically an error in mitosis. It's basically a mutation of a gene will encode checkpoint proteins, and the control mechanism will break down, and the cell will then undergo repeated and uncontrolled division. Now, a large mass called a tumour will then form, and the tumour will stop growing, and it will be encapsulated in a fibrous sheath, okay, and it will not travel to another location. Now, if it continues to grow, this is when it's called malignant, and this is whenever it gets serious. So a malignant tumour is the basis of what cancer is. It's a cell from a primary tumour is shed, then carried around the body by the circulatory system, producing metastases, which is secondary tumours. Now, the treatments for cancer are different. So you can get different processes which can be targeted by using anti-cancer drugs. So, for an example, you can have DNA unzipping, which is inhibited by adriamycin and cytoxin, where two strands of DNA cannot be exposed and DNA replication will not take place, and therefore the uncontrolled division will then stop. You'll also be able to use the synthesis of nucleotides, inhibit that using 5-fluorouracil and methotrexate. And you can also have the formation of the mitotic spindle being inhibited by taxol and vincristin without the spindle chromatids, and therefore they cannot separate. So, interphase, the non-dividing stage. We already talked about it briefly, but essentially, non-dividing stage, DNA is already replicated, so there's no chromosomes visible. So if you get diagrams where you know which one's interphase, you need to be able. This is how you basically identify. You'll see a nuclear membrane, 2 centrioles, and a nucleolus, but no chromosomes. Prophase. So prophase comes after interphase, and this is when the DNA will coil and condense it into compact shape. So each of the chromosomes will become stainable, and it still has 2 chromatids attached to a centromere. Now you'll also see a nuclear membrane, just like an interphase, but you'll see centrioles moving apart towards an opposite pole, so like north and south for an example. And the microtubules will assemble around the centriole, and no longer you will see a nucleolus. Now metaphase. We did talk about this briefly once before. Basically, nuclear membrane breaks down, you won't be able to see that, and the centrioles will reach their opposite pole at this point, with the microtubules extending between them, forming spindle fibers. Now the chromosomes will move to the equator, so that's basically horizontal line in the middle. So it moves to the equator of the spindle, and each one becomes attached to the spindle by the centromere. Then anaphase will occur, and this is when the centromeres will divide. So the centromere goes, and basically the 2 chromatids are separated. So the centromeres will divide, and the movement and contraction of spindle fibers pull these sister chromatids apart towards the opposite poles of the cells where the centrioles are. So now telophase. Telophase is when each chromatid becomes a separate structure, and then is then called a chromosome. So they're on their own. And the nuclear membrane will then reform around each group of chromosomes. So there'll be 2 different groups. The chromosomes will then unwind into chromatin, and the nucleoli will form in each new nucleus. So basically you'll start to see coming back to all the features you had in interphase. The spindle fibers will then go away, and the centrioles will divide into 2. Now we'll go cytokinesis. This happens differently in animal cells and plant cells. So cytokinesis in an animal cell, you'll have the cell dividing, forming 2 daughter cells which have an identical chromosome makeup that the parent cell had. If it doesn't, then a multinucleate cell is produced. Now in animal cells, you'll have a cleavage furrow forming, and the cell surface membrane will invaginate. Now then, microfilaments will attach inside the cell membrane around the centre, drawing the membrane inwards, creating a furrow, and the membrane will be pulled until the cell is divided. This differs from in a plant cell, as in plant cells, microtubules will remain in the central area, guiding the Golgi vesicles to the equator. These vesicles will enlarge, fusing together from the cell plate, and inside each vesicle, the middle lamella will form cellulose either side, forming the new cell walls, because we know cellulose forms cell walls. So these membranes of vesicles form new cell membranes, and small gaps are left between the vesicles, and these form plasmodesmata. Now, it's quite difficult to find cells going under mitosis because it only occurs at a specific part of the shoot tip, and the division is has a very small number of cells. The process can be easier using a low power microscope, identifying the zone division. The cells are smaller and cuboidal, and then they have a relatively large nucleus and the cell volume ratios. It's quite significant about mitosis and its role in asexual reproduction, as daughter cells will have the same genes, and as an organism develops, the cells will differentiate skin cells or nerve cells. Now, in asexual reproduction, the cell's parent will divide, producing cells forming offspring production of genetically identical individuals, which will suit identical offspring. Here's your difference between mitosis and meiosis. Basically, in mitosis you have 1 division of 2 daughter cells, and in meiosis you have 2 divisions. Basically, parent cell, you'll have a diploid as opposed to a haploid. The daughter will have the cell, same number of chromosomes as the parent cell. Meiosis, parent cell is diploid, daughter is haploid. Then you have your homologous chromosomes not associating during prophase, but the homologous chromosome pair will form bivalence during prophase 1. There's no chiasmata in mitosis, whereas it is in meiosis, and of course the daughter cells will genetically differ in meiosis as opposed to mitosis. Now meiosis, you don't really know a lot about it yet, but we will be back tomorrow for our meiosis video, so hopefully you can enjoy that when it comes out. Anyway, I have been Dylan from C.A.R. Entertainment. Hope you enjoyed this video, and I will see you next time.